So we're going to turn to Andreas Mayer, who is our early career scientist, but interesting, he's at the Princeton Integrative Genomics Group, Lewis Sigler Genomics Group, but I don't want to draw a direct line to the fact that he's in the iReceptor Plus seminar series, but he's just been taking a new position as one of the newest group leaders at the University College London UCL Division of Infection and Immunity. So it's a nice timing for have Andreas talk to us. And he's going to tell us about structure and immune receptor epitope maps, and take it from there, please, Andreas. But you're muted.
Okay, so now I'm no longer muted, am I?
Yeah, you're good. You're good.
Okay, excellent. Yeah, so thank you so much for the invitation to speak in the iReceptor Plus seminar series. And I'm very excited about speaking in the seminar series today because, as has just been mentioned, I recently moved and opened up my own group at University College London, focusing on quantitative immunology. But much of the work that I'll be talking about today was done when I was an independent postdoctoral research fellow at Princeton University, together with an amazing set of collaborators at Princeton, in particular, including Curtis Callan, and also including an excellent student, Andrew Pio, and another collaborator, Ned Wingreen, all at Princeton. And so the previous talk, I think quite nicely has set up a lot of what I'll be describing in this talk, and the sense that this talk is also going to be about antigen specificity. But here in this talk, it's going to be in the context of T cells. So as we've already seen in the previous talk, receptor-epitope binding is really the key that enables the adaptive immune system to respond specifically to different kinds of targets. And so just sort of very schematically, T cells interact with antigen presenting cells, and they do so specifically via a transmembrane receptor, the T cell receptor, that interacts with a peptide MHC complex that can be presented on the surface of these antigen presenting cells. And the hypervariable loops of the T cell receptor are in most close physical proximity to the peptide MHC complex. And those are these loops are genetically encoded through a genetic recombination machinery. So those are highly variable, which allows different T cells to have specificities to different kind of ligands. And a better understanding of the mapping between the various T cell receptors and the peptide MHC complexes that it can recognize is, I think, one of the major goals in immunology, to go from sort of all the receptor sequencing data that we in the AIRR community field care a lot about, towards something more functional, which is what are all the different epitopes that actually get recognized by these receptors, and ultimately answer questions such as questions about cross-reactivity. How does our history of prior exposures make us more or less susceptible to infection through T cells, for example, that can cross-react with a different peptide MHC complex, but also for questions such as immunodominance. So how does the genetic recombination machinery give rise to an uneven coverage of different peptide MHC complexes? We know that certain receptors are generated much more readily than others. And so if on top of that, we knew how receptors mapped into sort of groups of receptors that target the same peptide MHC complex, maybe we could make some headway into defining sort of landscapes of immunodominance beyond the rules of peptide MHC presentation. And so the reason I think that there's now some hope at making progress on the problem of mapping T cell receptors to peptide MHC complexes is because of a great increase in our experimental ability to measure these receptor ligands interactions in a high throughput manner using sequencing. And so there has been, starting with the sort of two pioneering studies by Paul Farmer's lab and Mark Davis' lab, were published about 5 years ago now, an increasing number of studies that have produced receptor epitope maps for T cells that go from that sort of old structural understanding where you have a single T cell receptor in complex with a single peptide MHC complex to sort of, you take a blood sample, you sort out, for example, using tetramers, the specific T cells from the sample, and then you sequence the hypervariable T cell receptor regions of these receptors. And so what you get from that is a relatively large set of T cell receptors that are all specific to the same target. And the specific visualization of this kind of data from a study from the Paul Farmer's lab is displayed down here. So in that visualization, what we show here is on below the diagonal the distance of the CDR3 alpha sequences, and above the diagonal the pairwise distances of all the CDR3 beta sequences. And what is quite apparent is that there's a lot of structure where a sequence similarity to an extent predicts antigen specificity. And so one common approach coming from the computational field has been to try and sort of use that sequence similarity to do clustering, to look at sort of the various sequence logos of these various clusters, to define what are the sequence determinants of the set of T cell receptors that bind to one specific target. And one thing that is sort of interesting is that there's two kinds of degeneracy. One is a local degeneracy where there's these groups of receptors that vary slightly in sequence, but then there's also global degeneracy, that there's other groups of receptors that bind to the same target and that have some locally close similar sequences, but also globally actually don't look alike like these other sequences. And I think that's a sort of an interesting insight in how one might think about the way that one should design inference methods for predicting antigen specificity. But here in today's talk, I want to make the case for a slightly different approach and for a slightly different question that we feel might be even more interesting if we can make headway on it, which is the question of, are there any ways to actually scale this up to the repertoire scale? So if you're thinking about a full immune repertoire, then ultimately we need to deal with the sort of very large dimensionality of this ligand receptor interaction, where we can have on the order of 10 to 12 different peptide MHC complexes, and the genetic recombination machinery can generate up to 10 to the 61 different alpha beta receptors, as it has been estimated. So then if we want to understand this at scale, clearly we will need to have data on receptors that are sequenced to lots of different peptides, and we increasingly have that, and specifically, and now there has been relatively large efforts in surveying sets of antigen-specific receptors in response to SARS-CoV-2. But we also need to then use this new kind of data to ask whether there are any generalizable rules for this mapping. So to go beyond the question of what are the specific sequence pattern in the set of receptors that recognize one specific peptide MHC complex, and ask more generally whether there's anything predictive around the rules of which kind of receptors match to which peptides that might generalize across different peptides. And we have started to think about this question a bit, and today I want to share with you some of the sort of preliminary progress that we've made on this this question. And so we sort of went back to the very basics to ask what can we learn from immune receptor epitope maps, where the basic setup is that you take a blood sample where there's a certain set of T cells with a certain set of receptors. And then through some functional criterion, you select out some subset of all the T cells that are specific to a certain target. And so the task then on the computational level is to detect selection in these receptor sequence repertoires. And beyond that, to then go and infer the statistical rules of selection, so the imprint of selection in these receptor sequence repertoires. And now I will share with you how we think about doing that and what sort of mathematical framework. So if we go back to sort of classical population genetics, how do we check selection there? One of the classical measures in population genetics is the DN-DS ratio. So this is comparing the rate of non-synonymous to synonymous substitutions along an evolutionary lineage. And the basic insight here is that if one has functional selection that's happening on a protein level, that should only care about the amino acid structure. So synonymous substitutions should be neutral with respect to functional selection. And so the question is, how can we adapt this to the current context of the selection that is happening on the set of T cell receptors? With the difference being that in this context, we have selection on standing variation. So for T cells, there's no hypermutation, such as for B cells, but instead there's a large standing variation that is created by the recombination machinery. And we select out certain receptors out of that prior pool of all the cells that are in a blood sample. And so the question is, how can we adapt this basic insight that functional selection acts coherently on the level of amino acid sequences to this context where we're trying to detect the imprint of selection that is happening on standing variation. And our proposal is to use coincidence counting. So to count the number of nucleotide sequences within a sample that map onto the same amino acid sequence. And so to illustrate this, I want to introduce this graph-based visualization where every vertex is a nucleotide sequence and different vertices are connected by edges if the two nucleotide sequences code for the same amino acid sequence. So then in this visualization, the probability of coincidence is given by this formula here. So it's related to the number of edges divided by basically the square of the number of vertices. And we can ask, how does that measure behave under different kinds of sampling? So we can, first of all, convince ourselves that if we just select a random subset of all nucleotide sequences, so if we just make a random sample of all the nucleotide sequences from the blood, well, then this measure actually doesn't change. So statistically speaking, the probability of coincidence will be the same if we have a smaller sample than a larger sample. So that's a good property. So what about functional selection? So if we select on antigen specificity, that would mean is that whenever we have a nucleotide sequence that maps onto a certain amino acid sequence, and then that is specific, we should also select all other nucleotide sequences that map to that same amino acid sequence because they're also specific. So if instead we're selecting random amino acid sequences, then the probability of coincidence goes up. And this can be understood quite intuitively here, that now instead of selecting sort of individual vertices at random, now we're selecting connected components, so these clusters at random, and that will increase the proportion of edges that we retain relative to the number of vertices that we retain. And so now, how can we apply this coincidence counting framework to data? To do so, we can basically look again at this visualization that I already introduced earlier that looks at all the pairwise distances between CDR3 alpha and CDR3 beta sequences. And we can slightly generalize the idea of coincidence counting to also look at near coincidences. So we define a generalized probability of coincidence at a certain sequence distance delta as the probability that if I pick two random different nucleotide sequences from a sample, that they're going to be at a certain edit distance. And that corresponds to the distribution of the off-diagonal elements of this matrix. And so we can do this both for the CDR3 beta sequence and for the CDR3 alpha sequence, or for the paired distance from both sequences together. And what is very striking is that if we compare the distribution of these coincidence frequencies in peptide-specific sets of receptor sequences to a background set that is just a straight DAT sample, that we see a very clear and very remarkable imprint of selection. So a very large selection strength that is implied by this level of convergence that we measure here using this coincidence counting framework. And we can then ask on sort of theoretical level, if we have a certain distribution of sequences that we start with, and we select certain sequences with probabilities Q that depend on sigma. So that's is some model of how specificity depends on sequence. And then we get the distribution of the sequences in the selected set, how what kind of properties of Q determine the shape of these coincidence distributions. And what we were able to show is that there's some simple insight of sort of doing the math of how coincidence measures change under selection, which is given by this formula here, where the insight is that near coincidence ratios, so the ratio of coincidences within the selected set to the set that you're selecting from, is telling you about how selection covaries with sequence. So here, this is an average of the selection factor across all sequences that are at a certain distance. So they learn directly something about the covariances between selecting a sequence and selecting another sequence at a certain distance by looking at the ratio of coincidence frequencies. And we get an even simpler result when we look at selection for specific binding, where the Q is sort of binary, so you either bind or you don't bind. And then what we find is that the coincidence ratio is at a certain distance delta is equal to the coincidence ratio for precise coincidences, where we share exactly the same amino acid sequence, times the fraction of sequences that is specific at a certain distance. So this is giving access to a very interesting property. So this is this quantity here is the conditional probability that if you know that a certain sequence is specific and you make a certain number of amino acid substitutions to that sequence, that it's still going to be specific to the same target. And so applying this now to this experimental data, plotting this ratio of the coincidence frequency within specific data relative to a background, what we find is a remarkably common signature of selection across a multitude of data sets where epitope-specific cells are defined in different ways. For example, in the DASH data, this is sort of something where tetramer sorting has been used. On the other hand, here there's a data set where a set of responding T cell receptors were defined by looking at the longitudinal dynamics of what are the kind of receptors that come up in response to a SARS-CoV-2 infection. And what we find is that across these very different modalities, all the different data is well described by a common functional form of how these coincidence ratios fall off with sequence distance, which is sort of an exponential decay by factors of roughly 10 per amino acid. And that suggests that roughly 10% of sequence neighbors maintain the same specificity. And so this puts a clear probabilistic statement behind the notion that on the one side, people that have tried to sort of use mutagenesis to look at whether neighboring receptors also bind often find that even small changes in the CDF3 region can actually abrogate recognition. But at the same time, it also is compatible with the usefulness on sort of a broader scale of clustering methods, because if the conditional probability of a neighboring sequence being specific is 10%, that's still much higher than the 1 out of 10 to the 5 or 1 out of 10 to the 6 maybe of the background frequency of any odd receptor being specific. And so one way that one can refine this is to ask, out of all these neighboring sequences, can we actually predict which of them are more likely to bind? And it turns out that we can use this coincidence counting framework to set up a sort of rigorous composite likelihood maximization framework where we can learn model parameters of a distance metric that maximize the composite likelihood of observing specific sequence pairs. And what we found in doing so is that there's physical chemical rules about substitutions, what kind of amino acids are substituted with each other, and at what positions within the hypervariable regions that are predictive of specific binding. And the application of these sort of more fine-grained distance metrics hopefully will be to use these statistical rules together with the knowledge of the recombination statistics to map precursor frequency landscapes. But before concluding, I want to highlight another interesting consequence of the sheer level of selection that is implied by antigen by receptors being sorted for antigen specificity. So this is in a background where you sort of have independent pairing of both alpha and beta chains, would predict that the probability that two receptors share precisely the same alpha and beta chain is only on the order of 10 to the minus 11. So it's very rare event. And despite that, in actual data, we see a very large amount of convergence. So similar to what we've seen for the BCRs in the last talk during the discussion, there's a very high level of convergence in receptors have been sorted for antigen specificity. And so if you think about a memory repertoire as being a mixture of epitope-specific groups, you can actually derive a decomposition theorem where the coincidence probability in such a mixture distribution is given by the sum of two terms, the coincidence within the receptors that are specific to a certain target, and the coincidence probability between targets. And if the number of targets is relatively limited, which might be due to sort of immunodominance, there's a prediction that we should actually see an imprint of this selection even in unsorted repertoires. And this is exactly what we found. So this is looking at a large data set of straight PBMC T cell receptors. We find that there's an enrichment of coinciding receptors beyond background in such a data set. And we can convince ourselves that this is actually a signature of antigen-driven selection. So one way of doing that is to show that actually within naive cells, there's less such enrichment. And moreover, and maybe even more strikingly, if we compare across individuals that differ by HLA type, we see that they also show less of that enrichment, showing that indeed the enrichment relative to the sort of independent background is driven by groups of cells that bind to the same peptide energy group. And so with that, I want to thank the organizers again for the invitation to speak today, and I'm looking forward to the questions and discussion.
Well, thanks a lot, Andreas. A lot to think about. Is this like you start with DNDS and evolutionary biologists use that all the time? And I mean, could I take this back and look at selection on opsin genes in guppies, or is this somehow the fitness function is epitope specificity, or?
Yeah, so here the fitness function is basically epitope specificity. And I think the way that we are thinking about is that it is basically a population genetics problem, right? You have a set of receptors that you're starting from, and you're somehow selecting them in some way by doing an experiment, and you get some receptors out. And so the question has been for us how we can adapt something like DNDS to the context of T-cell receptors. Yeah.
Okay. Not seeing.
Andreas, it's really interesting. So I just had a question about TCR versus BCR, right? So TCRs are recognizing, a peptide in the context of, MHC presentation, right? So do you think a similar approach clustering wise would work for BCRs? Because, so one of the problems BCR prediction people think is that you have structural convergence that is not necessarily sequence convergence, right? So you have different sequence solutions to the same structural problem. Do you think that this might be, flexible enough to accommodate those kinds of things, or not? I mean, it's tough for you to answer without data, but speculate a little bit.
Yeah, I think it's a very interesting question. I mean, I think the one thing that is interesting that even in the TCR context, if you look at these sort of maps, there seems to be different structural solutions. I think it's quite likely that these sort of different sequence clusters actually correspond also to different ways of binding the same epitope. So in that sense, I would hope that this similar framework can also apply to B-cell receptors, because even in the T-cell context, despite the sort of more restrictive binding interface because of the MHC restriction, it still seems that there's different structural solutions. I think the one thing that is really interesting about the B-cell context is that there one has sort of a mixture of selection on standing variation on all the sort of different close to germline receptors that VDJ recombination produces, together with somatic hypermutation. So one would need to generalize our sort of mathematical framework to include both. So both the selection that happens following hypermutation and the selection that happens on the standing variation. And that's one of the directions that we're thinking about going in. I'm not yet quite clear how we're going to do that, but I'm sure there's going to be ways to adjust the framework.
Yeah, I mean, this is super interesting to me too, because so we've been, we do antibody discovery primarily, and we've been moving from like these lower throughput single cell PCR things to stuff like 10x, right? And so that removes a huge bottleneck, but it doesn't actually remove a bottleneck. It just moves the bottleneck down one step, which is now we have to make them all and test them all functionally, right? So if we have thousands of antibodies, we can only make hundreds. How do we down select from that using just the sequence and maybe a little bit of other functional data to give us an idea of which antibodies we should be expressing? And if we can do stuff like this and say, like, you should only make one or two from this big cluster because they're all going to behave similarly, that's a really, really useful tool for sure.
Yeah, I think that's a big hope. And I think the other thing that is kind of exciting in that area is that, I mean, on the one hand, as we've seen, like also even with like LibreSec, I mean, there's all these new approaches of generating sort of bigger and bigger amounts of data on antigen specificity. But then at the same time, given that we actually see these signatures of selection, even in unsorted repertoires, as soon as we have sort of paired chain repertoires, we can, that sort of gives us another way of getting at pairs of sequences that are specific to the similar target. So hopefully we'll sort of pretty soon get big enough data sets that we can actually do some amount of machine learning to learn some of the rules and to really help then in a practical sense sort of refine the kind of T-cell receptors or B-cell receptors that you would test experimentally.
Yeah, that'd be like the perfect virtuous cycle, right? So we generate more data from single cell data sets, which you can help us analyze with these kinds of models, but then that data also makes the models better, and you keep kind of keeping that wheel keeps spinning, right? Yeah, that'd be great.
Yeah.
Well, great. Thanks again for two great talks to our speakers. The bad news is in May and June, we don't have seminars, I don't think, or next one because of meetings. Our next seminar series will be on July 28th with Jenny Jiang from University of Pennsylvania. And with that, again, thanks for all the participants and the speakers, and we'll see everybody soon at either the AIR meetings or other types of, come back to the seminar series. Thank you.
Thank you again for putting it all together.
Yeah, great.
Thanks, everyone.
Thank you very much. That was great.
So Felix. Felix.
Yeah.
Can you stop the recording?